In the last video, we talked about the process of transcription, going from a DNA molecule, the information on a DNA template strand, and coding it into an RNA molecule to create a copy of the original information. So everywhere where there's a T, we put an A. The enzyme that accomplished that is called RNA polymerase. RNA polymerase makes this molecule, and then once we had finished the 3 steps of transcription, initiation, elongation, and termination, we went through a process called RNA processing, wherein the non-coding regions of the mRNA message were snipped out. We call those introns. We cut the introns out and we spliced together what remained, the exons. The exons are the segments of the mRNA molecule that actually do code for things. We also slapped a 5' cap and a 3' poly A tail on the end in order to protect the mRNA message on its journey from the nucleus over to the rough endoplasmic reticulum, where it's going to bump into some ribosomes. So finally we can see how we get a polypeptide from the information in DNA. That process requires going from nucleic acid language to amino acid language. That means we're going to need some kind of molecular interpreter. And our molecular interpreter is another kind of RNA molecule, which is called transfer RNA or tRNA. So transfer RNA is what actually reads this message 3 letters at a time and then knows which amino acid to put in this polypeptide chain. For review, a polypeptide is just a chain of amino acids. There are 20 different amino acids, and the purpose of DNA is to hold the information for knowing what order to put the amino acids in for each protein. So DNA tells you how to make proteins, and that's what we are going to do now. We also need some workspace, a workshop, in order to build this polypeptide. That is the ribosomes. The ribosomes are going to be our workbench to actually get this process of translation done. And, of course, we're going to need the message. We're going to need our messenger RNA that we just made in the last step. So those are at least the 3 things that we are going to require. Now, there are about 30,000 different proteins inside each one of your cells. Sometimes this number gets adjusted up or down, but somewhere in the scope of about 30,000 different proteins. And that sounds like a lot of different proteins. But if you think about it, it's really not, because a protein is a polypeptide, and there are 20 different amino acids that you can put into a polypeptide chain. So let's say for the first amino acid, there's 20 possibilities, right? 20 possibilities of what could be the first amino acid in my chain. There's 20 possibilities for the second amino acid in my chain. That means that if I multiply these 2 together, I find that there are 400 different dipeptides that I can make. 2 amino acid long chains, 400 different dipeptides. If I want to make a tripeptide, right, I have another 20 possibilities for the third amino acid, which would bring the total number of possible tripeptides up to 8,000. And if I want to make a tetrapeptide, quadrupeptide, a 4 amino acid long polypeptide, then now it's going to be 160,000 different possible combinations of amino acids. Now, a functional protein isn't 4 amino acids long, it's thousands of amino acids long. So the number of possible combinations, the number of possible polypeptides that we could potentially make, it's virtually limitless. It's incalculable. It's a huge, huge number. So the fact that there's only 30,000 inside your cells should indicate to you that only certain combinations work. So this is what's important. We have to make sure that we get the information correct, that we translate correctly, in order to make sure that we don't make one of the many, many, many possible polypeptides that are non-functional in the body, or that are potentially dangerous inside the body. We need to get the message translated correctly. So how are we going to do that? Well, every 3 nucleotide letters in the mRNA message is referred to as a codon. A codon is essentially a word in nucleic acid language. If an mRNA message is 30,000 nucleotides long, it contains 10,000 codons, because every codon is just 3 of those letters right next to each other. So, unfortunately, we're going to have to do some actual translation work. You didn't know you were going to learn a new language today, but that is what we're going to do. We need to learn what the words of nucleic acid language are and what they mean in polypeptide language, in amino acid language. Now, the good news is that there are only 4 letters in nucleic acid language, and every word is exactly 3 letters long, which means you can actually figure out how many possible words there are, how many different types of codons there are. Each word 3 letters long, and there's only 4 letters to choose from. If you multiply this together, you find that there's 64 possible combinations of nucleotides, 64 possible codons. And every single one of those codons has a specific meaning. 3 of them are what we call stop codons. Stop codons. They tell the ribosome that it's done its job. It's already completed the polypeptide and it needs to stop. The remaining 61 codons are amino acid codons. So AUG tells you a specific amino acid. GCA tells you a specific amino acid. So we're translating from a language that only has 64 words in it. Hey, this is going to get a little bit easier than maybe we thought at the beginning. But there's good news. We're translating from a language with 64 words in it to a language with only 20 words in it, because there's only 20 amino acids. So even though there's 61 different codons, some of them are going to code for the exact same term. So in order to complete translation, we're going to need some sort of translation key to get us started. Enter the genetic code. The genetic code tells you what each one of these codons mean. So there's been a lot of complicated language so far. Let's bring it back down. This is our mRNA message that we just made. It's a transcript, a copy of the information inside one gene. Every 3 letters of this mRNA message is a codon. Every codon translates to a particular amino acid, or it tells the ribosome to stop translating here. So I'm going to pin our genetic code right up here, and then we're going to read from it in order to try to figure out what this mRNA message says. Our first letter is AUG. So if we look at that genetic code and we zoom in on the right part of the sheet, you'll see that AUG translates to an amino acid which is called methionine, and it's abbreviated MET. MET stands for methionine. You'll also notice that methionine is labeled as being the start codon. The start codon. It turns out that all polypeptides, all proteins, start with methionine. Just kind of a quirk of evolutionary history there. So that's our start codon. We begin with methionine. Next one, GCA. GCA. Let's take a look. Now on this chart, G is the bottom letter on the left-hand side, and C is the second letter going across the top. So if we zoom into that segment right here, we should be able to see, yep, right there, GCA gives us an amino acid called alanine, which is abbreviated as ALA. These 3-letter abbreviations are perfectly fine for your answers. So we went from methionine to alanine, and now we have UAA as our final codon. So let's find that UAA up on the genetic code. If you take a look right here on the genetic code, you'll find UAA, and UAA is one of our stop codons. A stop codon doesn't code for an amino acid. A stop codon tells the ribosome that translation is complete, and we've already finished this message here. So this is what our initial DNA template strand was actually trying to communicate. Methionine, alanine, stop. We made a dipeptide in this process, something that's just 2 amino acids long. Obviously, it gets a lot more complicated, but this is a good way to get started. Now try this part out for yourselves. I'm going to bring in an RNA message right here, whoop, and I want you to try to translate it using this genetic code right here. Remember, every 3 letters is a codon, and every codon has some translation. It'll either be an amino acid codon, or it will be a stop codon. Most of them will be amino acid codons. Now, carefully look at how this chart is organized. Every codon that's labeled in this top row of boxes starts with the letter U, and then in the second row down, that's all of the codons that start with the letter C. In the third one down, it's all of the codons that start with the letter A, and then in the fourth one down, it's all of the codons that start with the letter G. Then it's organized by columns. The second letter U, so all of the UUs are in the top right-hand corner, and then right next to them is all of the UCs. So find the first and second letters and find the correct box that you're looking for. And then once you have the correct box, it's relatively easy to find the codon that you're searching for. So take a minute now and try to translate this RNA message at your desk. I'll wait. So hopefully you got this as the correctly translated message. If you got it, pat yourself on the back. If you didn't get it, then you're going to want to practice this a few times. There will be some practice problems in the book that'll be available to you, and I'll see if I can make something else up that's just a series of practice problems with this idea.